Hi, so we're going to learn a little bit more about how to sequence the genetic code or the DNA of organisms, right? How do we go in and can actually read the nucleotide letters? Well, first we need to back up and remind ourselves about an important process that we've learned already, polymerase chain reaction or PCR. Recall that we can start from an initial fragment of DNA and we can make multiple copies of this by allowing the fragment to separate, to unzip, and then we can take advantage of our knowledge of replication and have a polymerase that functions at high temperatures because we can put it in these machines up here called the thermal cycler, which heats up the DNA so that it separates, it unzips, leaving those nucleotides now free to serve as a template. And the polymerase can then run along these free ends and can make new copies. So we started off with two black copies, we go to now a black and a blue copy, and we can then let it cool down and they come together, and then we can heat it back up and let the replication happen again, and you can get now new copies. So you go from now 2 copies to 4 copies. So each time it's duplicating, right? And then we go to 8 copies. And eventually, after lots of times, we have millions of double-stranded DNA molecules of that same segment. Once we have that product, we can now add something new to it. So in this new reaction, we now have all of that DNA, those millions of copies of DNA. We can add more ingredients to allow another type of chain reaction to occur. So we're going to add a DNA polymerase, we're going to add some primers like we had to add before, but now we're going to add, in addition to the regular nucleotides, right? Because you have to have lots of extra nucleotides so you can build all the extra DNA. And this is what's figured right here, just the dNTPs. But we're also going to add special types of nucleotides this time called that are called ddNTPs. And these are nucleotides that have a special fluorescent dye on them, and they also are nucleotides that don't allow the replication process to continue beyond them. So you put a few of these, in relatively speaking, to the regular nucleotides in this reaction tube. And then you're ready to start the reaction. Now I just want to give the overview of where we're trying to head. What we're trying to head to is that if you do this enough times where you randomly grab one of the fluorescent nucleotides, you should be able to read all of the different nucleotides along a DNA sequence. Let me explain this a little bit further. So again, we start off with a piece of DNA that has been separated because we heat it up. The primer links on, and then beyond the primer you start to build, right? You start to build from the 5' to 3' direction, and you add a nucleotide. And if you add a nucleotide and that first nucleotide happens to be one of the nucleotides that is fluorescently dyed, it'll stop right there and you don't add any more nucleotides. And so now you have that first nucleotide beyond the primer, you have it now dyed and it's ready to be read. And then you keep doing the reaction, and again, you're going to do this so that you get millions of copies. Well, if you do this enough times, randomly you're going to get it where sometimes the reaction will grab regular nucleotides and then proceed down and then hit a fluorescent nucleotide and stop. And so you do this enough times, and essentially you get enough copies where you get copies that are only 1 nucleotide beyond the primer long, 2 nucleotides beyond the primer long, 3 nucleotides, and so forth, all the way down the fragment of DNA that you initially carried out the PCR reaction on. So you do that, then you take all of those reactions and you send them through a small capillary gel. So this is just kind of like we talked about before with gels. DNA you can send through a gel if it has a charge on it, and as the DNA goes down through a gel, it separates based on its size. So the smallest pieces, in other words these pieces over here, will go the fastest through the gel, so they'll travel first through the gel, and the longer pieces will travel last through the gel. So as they're coming down the gel, there's a laser that can read that dyed ending nucleotide, that nucleotide that's a ddNTP that has that fluorescent dye on it, and it can read that, and the detector then can detect the peak and then translate that into the actual nucleotide, whether it's an A, a G, a C, or a T. Hi, so I found this animation I really liked. So here's that capillary, right, that the fragments of DNA are being drawn through this, the smallest ones first and the longest ones last. And as they run across that laser, the laser shines on that dyed nucleotide, and that's what determines what the read is, whether it's an A, G, C, or T. So that's the way I found this. I wanted you to take a look at that. I think that kind of completes the story of how the nucleotides are read. That then gets put on a computer. We can visually look at that at what's called a chromatograph, also what I showed up here. And therefore we have the laser detection of those fluorochromes, or of those nucleotides that have been dyed. And then that can then be written as a DNA code, right? GAT, AA, TCT, and so forth. And that information then can be used for a myriad of other analyses downstream. And that's how sequencing is done.